presenter here that we are going to introduce here shortly as soon as we have him available.
Oh, he's here already. He's able to share his screen whenever you're ready.
I'm here.
So just, yeah, Blaine's here.
Oh, hi, Blaine. Yay,
Hi.
that's what I was looking for. Fantastic. Mahalo nui for joining us today, Blaine. So real
No
quick,
problem.
I'd just like to introduce our next presenter. Blaine Luiz has been involved in rod pathology research since 2014 as a member of Dr. Lisa Keith's lab. He completed his MS in Tropical Conservation Biology and Environmental Science program at Hilo, where he studied the morphological characteristics of Ceratocystis leucohea and conducted the first disease screening of ohia with the pathogen. He is currently the coordinator for the ohia disease resistance program as a part of the Akaka Foundation for Tropical Forests, where he continues to work of screening ohia throughout Hawaii for natural resistance to rot. He hopes to one day return rod-resistant ohia trees to our forests and begin repairing the damage that rod has caused. So thank you so much, Blaine, for joining us today, sharing your work with all of us, and we look forward to learning more from you. So take it away.
Yes, thank you very much for having me today. As Hoku said, I am Blaine. I'm the coordinator for the ohia disease resistance program. And I'll be discussing our hunt for rod-resistant ohia genotypes, specifically building the foundation for an ohia disease resistance program to return ohia to our rod-impacted spaces. So I wanted to start by describing to you what plant pathologists, or how plant pathologists describe resistance. So resistance is the ability of an organism to exclude or overcome completely or in some degree the effect of a pathogen or other damaging factor. So this means it's an inherent quality of the organism, which makes it very useful because there's, other than finding it and producing enough of it, it doesn't really need any other input to keep away the diseases or the pests themselves. And so as non-native pests and pathogens continue to increasingly become prevalent in forests across the globe, disease programs and the development of disease resistance forest trees has become very popular and is a powerful tool for restoring populations impacted by diseases and other pests. Some examples of programs that have successfully produced disease or pest resistant forest trees are those for Western white pines, Port Orford cedar, and Sitka spruce. And even here in Hawaii, we have an example with koa, fusarium wilt resistant koa. That work being done by Nick Dudley and a bunch of other folks. So specifically thinking about rod and ohia, loss of ohia would be catastrophic to the ecological integrity of our native Hawaiian forests. Rod is a huge threat to that. And so thinking about how we can incorporate this blueprint that other programs, other resistance programs have made and have successfully charted the waters towards production of resistant plants for restoration and conservation. Are there, or better, where are our rod resistant ohia so we can do the same thing? To start answering those questions, we did a study back in 2016 looking at 128 plants from 4 different Hawaii Island varieties of ohia, Metrosiros polymorpha. Those were Encana, glabrima, polymorpha, and Noelii. And we screened them with Ceratocystis leucohea. And just to kind of show you the methods that we used, what we did was we made a little flap wound on the bottom left there and inserted a fungal colonized filter paper. So that's the little white thing in that wound there. And then we wrapped that up with a piece of plastic film to keep it all together. and try to inoculate the plants. And so once they're inoculated, they go into a growth chamber, which is a little chamber right there on the top right. It keeps a constant temperature and 12 hours of light. And so we had it at about 28 degrees Celsius and left them in there for 17 weeks and monitored these plants for rod symptoms and just kind of tracking them over time to see when they die, how quickly they die, and things like that. And so after 17 weeks, or throughout that 17-week process, as plants die, they get removed, or they were removed from the chamber, and they were debarked, as you can see in the lower right, which is considerably tricky, thinking about how small these plants are, and how small these stems are. It's very easy to snap them in half if you're not careful. So they were debarked using a small little scalpel or a small little knife, just to display the staining in there, which was a good indicator that the disease or the pathogen took. And staining was measured, as well as a bunch of other measurements. And so as a result of this experiment, about 82% of the ohia that were inoculated died. So that's definitely most of them. And most of them died within the first 49 days of inoculation. So again, this was running for 17 weeks, so 119 days, I believe. And a lot of them died right there in the first, third of the monitoring period. But we didn't see 100% of them die, which is great. We saw individuals of Encana, Nuelii, and glabrema still holding on after that 17-week period. Unfortunately, all the individuals of variety polymorpha died. There were none left at the end of the experiment. And I think that that is just a result of the conditions that these plants were in for the experiment. So as I was saying, it's a growth chamber, it's constant temperature. It's 28 degrees C in there, which is pretty warm. And considering what polymorpha is used to, I don't think that was the right environment for them, which really, again, kind of goes to show that when we think about disease and the disease process, there are three main factors involved, and that's the host, the pathogen, and the environment. And in this case, I think the environment really had something to do with how susceptible the variety polymorpha individuals were in this experiment. But nonetheless, it was hopeful to see that some survived, any survived at all. I think we were all in the beginning expecting the worst, and it was really good to see that things survived. Particularly out of all of them, Encana did the best and had the highest survivorship, which was about 37% of them surviving at the end. And so as of today in 2021, we still have some hanging on. So things continue to die after that 17-week process, but we still have about 5 Encana left and 1 Nuelii still alive. And that 1 Nuelii, I think, is a hybrid between an Encana and a Nuelii, because it's not exactly, it doesn't look exactly pure Nuelii. And plus they co-occur anyway out in nature, and so it wouldn't be surprising at all. So these survivors, as well as a bunch of other survivors from other inoculation experiments we've done throughout the years, are continuing, are still being monitored at the Institute of Pacific Islands Forestry, as depicted here. And our plans for these trees are to propagate them via cuttings or hopefully seeds when available, and to also get them in the ground and get them to grow as big as they can, and continue to monitor them for diseases or for symptoms. And so in light of this, these results, there's a big need for a long-term strategy for perpetuating Metrosideros or ohia as a dominant component of our native forests, especially under the threat of rod. And with the results we saw, I think it's easy enough to say that an expanded assessment of rod resistance in Metrosideros taxa, not just M. polymorpha, but all Metrosideros in Hawaii, to see how frequent it occurs in nature and just what the situation looks like out there. And so to meet this need, the Ohia Disease Resistance Program was formed in 2018. And so this is a partnership, an interdisciplinary group of a bunch of federal, state, and private entities, many of which are here today in this call. And we have a bunch of experts in various fields that are incredibly important for pulling off a complicated program such as a resistance program in fields such as plant pathology, ecology, plant genetics, tree improvement, and so on. And the mission of the ODRP is to perpetuate Ohia in the landscape without fear of rod, particularly through the identification, production of rod-resistant Ohia. To accomplish this mission, we're going to tackle this in two different phases. So the first phase is a research phase. And this phase is a very important first phase in the whole process, because this is the phase where we try to figure out everything from growing plants to screening the plants and making sure we refine everything and fine tune everything as best as we can. Because there's the potential for this program to screen tens of thousands of plants every year. If we can scale up to that size with our facilities, it's definitely possible. And to accomplish something like this in such a short amount of time, we'll need to have such a high throughput as that. So like I was saying, the two important steps of any resistance program are growing and of course screening the plants. So making sure that those two steps are really fine-tuned is going to be incredibly important. So for instance, for growing Ohia, we're planning a study to look at various different growth factors such as container sizes and figuring out kind of what combinations of things work best for producing Ohia to the appropriate inoculation size the fastest. We're also looking at our different inoculation methods. So right now we use that method that I explained to you before, which is the filter paper disc method, which Lisa Keith and her lab were the first to employ for this pathosystem. But there are a bunch of other different methods that are possible for inoculating Ohia, and we're currently working on that with Mark Hughes to figure out which method might be the best and produce the most consistent, accurate, and as close to what's happening out there in the woods as possible. And this is, again, is going to ensure that we have consistency in both our production and in the results that we have through inoculations, as well as increasing the efficiency of the program. So the next phase built upon that information is the operational screening phase. And so there's a bunch of different steps involved with this. So it starts with collecting, of course, the Ohia propagative material to grow and test. And so there's a bunch of different ways that we started doing that, which I'll explain in a little bit. But this is one of the most time-consuming parts of the whole process, because as you folks know, Ohia takes a long time to grow. It's on its own time. And so any way we can speed up that process is great, as well as just trying to get as much as we can from as many places as we can. Once the Ohia are to size, they'll be screened in a short-term greenhouse screening. So this is kind of the general weeding out step. A lot of the really susceptible ones will be screened out quickly, as evidenced by our first screening study. And any survivors that are left will be pushed along into the long-term monitoring step, which would involve field trials. So like I was saying with the survivors of the last study, getting them in the ground, seeing how they perform out in nature, and if those environmental conditions cause anything to change, and really just validating the short-term greenhouse screenings. So survivors that are still hanging on after all that abuse would, I think, very confidently be considered resistant to rod. And we could start establishing seed orchards with those field trials. We could convert them into seed orchards and hopefully start producing the first generation of rod resistant seeds. So that would be a really good, that will be the very important step of figuring out the heritability of this resistance, if it is and to what degree. And also taking a look at, especially if resistance isn't incredibly strong, if it tends to be weak or intermediate, we could work on breeding the survivors that are still around, and do successive generations of breeding to really increase the level of resistance and make it as strong as we can get it before releasing it back out into the wild. And so, like I mentioned with that first step, there's a lot of different ways to get ohia and to creatively get as much ohia as you possibly can. And this is one of the projects we have going on. So on Hawaii Island, there's a bunch of places where rod has gone through and taken out a lot of the forest. And however, there are still some that do hang on. And similarly, there are stands of otherwise healthy ohia kind of next to these sites. And both of these cases are really puzzling. And, you know, to kind of tackle that question, why are they here? If rod is so threatening to ohia, how come we still have things hanging on? Ohia is still hanging on. And so from a pathology standpoint, if rod has gone through, then nature has really done the first round of screening for us. So to take advantage of that, we have set up 4 different sites throughout Hilo and Puna on the Big Island, on Hawaii Island, and started employing this kind of paired approach study, looking at these areas with high rod impact. So just a handful of survivors left in an otherwise barren or ohia void landscape. And also in those same spots, looking at adjacent stands that are looking completely healthy and beautiful. And our goal is from these sites to take cuttings and seeds and grow those plants to start producing plants that we can test and really try to get an understanding of whether those ohia that are still living out there, whether they've been tested with rod, whether they've been infected, and just trying to validate what nature has done for us. So these are pictures of some of our crew members going out into the field and doing those collections. They have done an amazing job. They produce so much material for us, and I'm very happy to have them as a part of our crew. And as far as seeds and cuttings go, they'll go out in the day and they'll collect them and bring them back to the greenhouse. And especially if it's cuttings, they'll produce them, they'll process them in the same day, because the longer you wait, the viability of those cuttings greatly decreases. And they'll cut them up into smaller pieces and dip them in rooting hormone and plant them into trays of vermiculite. And after about 2 months or so, we'll check them for roots. And usually, because these trees are wild, a small portion of them do end up rooting. But from those plants, we can, if there's enough of them from a particular mother tree, we can screen them, or we will turn those plants into mother plants themselves at the greenhouse and try to fertilize them and take care of them and get them big and healthy to produce more cuttings. And along with this paired site approach, we have really started implementing technology to help us find these sites and develop our plots. So we've partnered with UH Hilo SDAV lab, and they're able to fly their drone and really get a bird's eye view of the landscape of some of these sites and help us to establish plots based on the parameters we've created for both high and low impact sites, and save our crews a bunch of time that they otherwise would spend on the ground trying to figure out and assess the site mortality. These folks can just fly over with the drone and get that real quick. It's very important. It's a very powerful tool for us to have and to be able to utilize for this. Similarly, we've partnered with Arizona State University, and they are working on developing technology to detect spectrochemical signatures for rod resistance using hyperspectral cameras. And so a lot of that I do not understand. It is beyond my understanding of, but it's a work in progress, and it's just starting, but there's a lot of promising results that are starting to occur. But once that technology is developed, and as it's developed, that also will be a very powerful tool for almost kind of pre-screening trees before we go out in the field and collect them. Again, saving our field crews a lot of time on the searching and collection phase. Also, because ohia is so special to a lot of people here in Hawaii, we want people to be involved, and we want the community to be involved with protecting their forests and producing the next generation of forests. And so we are super happy to have community involvement in our ODRP research, and we've partnered with the Leilani Estates community members and have established a paired site down there, looking at parts of the neighborhood that do have a lot of ohia still growing and still healthy, as in the picture right there, as well as, of course, the lower areas of Leilani where ohia have been devastated by rot, and there's not a lot of beautiful big ohia left. Thinking forward with this partnership, the goal is not only to collect ohia and remove it from Leilani, but eventually to return it back there. Once we've done our screening and done our tests, it's in the plan to return resistant ohia back to Leilani Estates and have them plant their ohia back in their community, which will also play into our long-term monitoring and field site establishment. Along with that partnership, we've partnered up with the Teaching Change program to produce ohia grow kits for a couple of schools here on Hawaii Island, so Lapahoihoi Community Public Charter School and Kohala Middle School. And these ohia grow kits are a couple of pots with some potting mix and fertilizer and a little packet of ohia seeds. And this is going to teach these students how to grow their own ohia and understand the process of going from an ohia seed to an ohia seedling and a plant, and at the same time being able to take measurements and collect data and learn the whole scientific process. And on the flip side, this is a good partnership because part of the seedlings that they produce, they'll get to keep and plant in their communities, but also part of those seedlings will come back to the ODRP for screening. So this is a good win-win situation for us. And so I've been really Hawaii Island centric this whole time, but I promise that we're not only focused on Hawaii Island. We do plan on testing ohia throughout the state. And we plan on partnering up with the ohia seed banks and utilizing those seed bank accessions for growing and screening ohia throughout the state. The ohia seed bank, the ohia seed banks are a powerful tool for this, as it's already a collection of genotypes from across the state, and so this saves our crews and our crews a lot of time and planning. And utilizing ROD threat level data produced by USGS and these provisional seed zones produced by Alex Loomis and NTBG and DOFA, we're able to prioritize seed zones under the highest threats of ROD so that we can try to target and identify the risk that ohia are under those seed zones. And currently we're holding off for now on really expanding into that area of research, but we do have some growing currently, and some of the taxa that we are growing are Metrocilus rugosa, Tremuloides, Micropus, Waialeale, and various M. polymorpha varieties from Oahu and Kauai. And so those are just some of the ways that we're producing or collecting the material for the program to begin screening. But from that point on, it gets into the actual screening part. So we plan on doing these inoculations at IPIF or PBARC, the Pacific Basin Agricultural Research Center in Hilo. Both are in Hilo, and doing those short-term inoculation studies there. And from that point, survivors would then be put out into field inoculation trials to verify those results. And again, as survivors continue to hang on, those survivors at the end of all that abuse will be put into seed orchards and hopefully producing the first generation of rod-resistant seeds. So the outcomes of all this work are the detection of both high susceptibility and highly resistant metrosideros genotypes. And both of those things are important because this allows us to understand the frequency and distribution of resistance throughout the landscape, as well as any differences between varieties and species. Also, highly susceptible genotypes are useful for our experiments because they can act as controls, making sure that our inoculations are working as intended. And of course, resistant ohia are the star of the show. That's the big kicker for all of this research, is being able to put out a rod-resistant ohia. And like I mentioned before about levels of resistance, if weak if the resistance is weak, we can invest in a breeding program to really try and boost up the levels that are present. And so we have accomplished in the 3 years that we've been operating a few things. So we've collected cuttings and seeds from over 200 trees throughout Hawaii Island alone, resulting in thousands of plants at various stages of growth. And this is actually our first target for our program. And so we just accomplished that last year. And so we're looking to expand beyond that 200 point and really increase the throughput of the program. This inventory that we have also includes ohia from 5 different metrosideros species and 5 varieties of metrosideros polymorpha. Again, really hoping to tackle what the distribution of rod resistance looks like throughout Hawaii and throughout all the metrosideros taxa. We also produced 2 peer-reviewed journal articles on the Stratocystis leuco-ohia research involving the culturing of Stratocystis leuco-ohia for inoculation, for inoculations, and as well as the initial inoculation study. And we also have started producing more outreach material. So we created a physical brochure and online content to highlight our projects. And I will share that website with you all at the end. Oh.
Great. And Blaine, I just want to interject. You have a few more minutes, and there's some questions in the chat box for you as well. So I'll go through that if you want to wrap it up soon.
Okay. Yeah. I have one more slide.
Perfect.
So looking forward from all this, we're currently in the collection and the growing phase. We have yet to start screening things yet, but we're getting close. We hope to start screening ohia this year, dependent on the results of our inoculation methods experiments. And we hope to be producing results real soon from our collections. We also will begin outplanting ohia that have survived from experiments in the past to really push some of our older survivor ohia into the next stage of this research. We plan on expanding our growing and screening capacities to facilitate a higher throughput system, and also expand on our seed and cutting efforts as well. And of course, we continue to test our methods and continue operationalizing our programs so that we can get the best results as quickly as possible for you all. So as a takeaway, I just want to say that creating a supply of rod-resistant ohia is complex, and it takes a long time. It can take a long time. It involves plant pathology, plant genetics, tree improvement, the whole kit and caboodle. And so it's going to take time to achieve really truly resistant plants that can survive in nature. But despite the fact that it can take years and maybe even decades to achieve, the fact that we have some ohia that have survived inoculations is really hopeful. And I think it suggests that there could be natural resistance out there, and it's worth studying. And with the nature of how resistance is, it may be our best tool for keeping ohia in our native forests. So with that, I'd like to say thank you for listening, And if you have any questions, you can contact me or I'll answer them in the chat. And you can see our website listed there.
Mahalo, Blaine. I just wanted to bring up, since we're in the question and answer phases, I was just going to bring up what's in the chat box. I think Christy had mentioned that, thank you very much for all your hard work on this. And there was a school in Waimea that mapped all of their ohia in their community, and they were interested in planting more. So I think we're going to try to link you guys up with them, maybe to work with them to some degree, and maybe taking a look at what you guys did with Teaching Change. To get them involved. So we will reach out to you on that front. I believe Lahela had asked, again, mahalo nui for everything. What is the size of the ODRP staff and what are the titles? Either supervisor, field staff, outreach, and where is the funding coming from?
For sure, yeah. So right now our staff is, so I'm the coordinator. I'm full-time on this project. All of our staff is funded through Akaka Foundation, which is funded by grants. And so we also have some field crews. There are about 4 folks under us working on ODRP. And the crews are really instrumental in going out and doing the field work and allowing me to do all the administrative and the planning. And a lot of the work that gets done in ODRP is through the help of our partner agencies. So the Forest Service, the Ag Research Service, the DOFA, and a bunch of our other partners have been really helpful. Yeah, that's pretty much that. I hope I answered your questions.
Yeah, sounds like it. Thank you, Blaine. As of now, there's nothing else in the chat box. If anybody else has any other questions, please feel free to put it in there. If not, Blaine, you can stop sharing your screen and we're gonna do a little quick